Hello and welcome! Ever wondered how scientists find a single, specific gene hidden within the massive library of our DNA? It's like trying to find one specific sentence in an entire library of books. The incredible tool they use for this is called a DNA probe. Today, we're going to explore what a DNA probe is and how this molecular detective gets its job done. A DNA probe is essentially a short, single-stranded piece of DNA or RNA that has been created in a laboratory. The most important feature of this probe is its sequence of genetic letters, its As, TS, CS, and GS. This sequence is specifically designed to be complementary, or a perfect match, to the DNA sequence of the gene or region of interest. Think of it like a keycut for a very specific lock. To make the probe visible, scientists attach a special tag or label to it. This label can be a radioactive atom or, more commonly today, a fluorescent molecule that glows brightly under a specific type of light. This tag is what allows us to see if the probe has found its target. The process of using a probe is called hybridization. First, you take the DNA sample you want to test, for example, from a patient's blood. This DNA exists as a double helix, with two intertwined strands. To allow the probe to work, these two strands are separated, usually by heating them. This is called denaturation. Next, the labeled DNA probes are introduced into the sample. The probe will now search through all the single-stranded DNA. Because of the rules of base pairing, where it only binds with T and G only binds with C, the probe will ignore all the DNA sequences that don't match it. However, when it finds its exact complementary sequence, the target gene, it will bind to it tightly. Once this binding, or hybridization, occurs, scientists can detect the probe signal. If the fluorescent tag starts to glow, it confirms that the specific gene or DNA sequence is present in the sample. This technology is incredibly versatile and has many critical applications. For instance, DNA probes are used to diagnose genetic disorders like cystic fibrosis, identify infectious diseases by detecting the DNA of a virus or bacterium, and are a cornerstone of forensic science in creating DNA fingerprints to link suspects to crime scenes. It's a powerful and precise tool for navigating the complex world of our genes. Thank you so much for watching. If you found this explanation clear and helpful, please show your support by liking this video and subscribing to the channel. It really helps us continue making educational content for you. We'll see you in the next video.